Welcome you all to the next class on Inorganic Chemistry of Life, Principles and Perspectives. So far we have tried to more or less complete the syllabus that is assigned to this. So, therefore, what I thought was, since it is a course, I better do some kind of a revision. So, this class, maybe one or two more classes, I will be revising the whole aspects that I have talked about till now in this particular course. Of course, highlights. Obviously, I cannot speak every aspect that I have talked for the past 40, 50 classes or so. Secondly, after completing that I will start getting into the tutorial parts, where I will explain some questions. I will also address their answers, those kind of things. So, with that the course will come to a conclusion. Okay, look at I started with this particular slide in the beginning with a title that how do I arrive at this particular kind of a title. As I told you, it is started with in the early 60s to late 60s etc. as inorganic biochemistry, where biochemists have identified some role of inorganic elements here and there. Therefore, they thought it is the inorganic biochemistry. Then later on, the inorganic chemists have come into picture and started looking at this with more details from the perspective of the inorganic chemistry, and that has they have named it as the bio-inorganic chemistry. And so, by that time the inorganic chemists have taken care of all this. Then not only inorganic chemists, the biochemists, the inorganic chemists, there are a lot of physical chemists, organic chemists have jumped into and started trying to make all kinds of studies, spectral microscopy studies, comparisons, model studies, all these, and found that the ions of the inorganic ions have a role across the biological systems. So, therefore, this has taken a nomenclature in this particular century as the biological inorganic chemistry. So, therefore, I thought what I thought was that I take the approach of the biological inorganic chemistry, but add a few aspects to make it inorganic chemistry of life. Is that where I have taken into consideration the aspects corresponding to the inorganic elements, inorganic compounds in the medicine, inorganic elements, inorganic compounds in diagnosis and therapy, etc. So, therefore, I have not only treated with the introduction followed by enzymes, but also have treated with these kind of things, as you can clearly see on the next slide. Very clearly. So, I have already taken you the introduction. In the introduction, I have covered a lot of aspects, elements in biology, which of the elements are responsible for biological processes and why, all these kinds of things, and their coordination chemistry, because they being the inorganic ions, all the coordination chemistry characteristics, how do they support this particular aspect, and simplistic coordination chemistry features also I have talked to you. Then I have talked to you biomolecules, introduced you biomolecules like proteins, nucleic acids, etc., all of those, and some relevances like protein synthesis. And there are a host of techniques from spectroscopy, from analytical spectroscopy, microscopy, lifetime, all these kind of things. So, I have tried to give you glimpses of all this. So, therefore, the introduction that I have given you was a combination of the elements in the biological systems, their coordination chemistry aspects, biomolecules, and the techniques. Then I have explained you a few classes on the biological inorganic chemistry of alkaline earth ions. Anyhow, I am going to give the highlights of all of these again now in this and next one or two classes, as I promised, and then enzymes, then followed by that about 30 plus classes or so I have spent on enzymes based on the transition metal ions. And then we have looked at some environmental kind of detoxification, which is called the mercury detoxification, called mercury reductase. Okay, that enzyme also we have looked at. Okay, then we looked at the enzyme based non-metallic element, which is selenium. The selenium in place of the cysteine sulfur, instead of that a cysteine without sulfur, but in the presence of selenium, selenocysteine, and that also we have looked at. And then towards the end, very recent few hours I have spent on inorganics in medicine, inorganics in the disease diagnosis and therapy. Okay. Now, I will go through some highlights of each of these from now onwards, a couple of hours of or couple of classes that I will be doing that. So, please try to be more attentive for these kind of a highlights, which of course I taught earlier, but this will be in the highlight fashion. Okay, so beginning, the first thing that I have talked to you is about the looking at the composition of the human body. So, the human body composition, if you look at that, there are elements which are called the bulk. The bulk elements are carbon, oxygen, nitrogen, hydrogen. If you take these carbon, hydrogen, oxygen, nitrogen together, is this called big 4, and this will constitute to something like a 96% of the weight of a body. So, this is we refer in the fatty. You see in the title fatty, fatty goes to that. Then you see some major elements like calcium, sodium, potassium, magnesium, all these things, and some few other things like phosphorus, chloride, sulfur, which would lead to the anion, which will lead to the cations. So, the cationic and anionic, these ones, and this is around 3.9. So, 96 plus 3.9 is 99.9. And these things are the ones which you know the bones and all, everything is the one which gives the shape to the body. That what I am referring to, strong. So, these are the elements which give the strong. And what is health? So, it is like a body, then you have a bones, and then you have a health. So, health is given by these trace elements. The trace elements itself is 0.1% of the total body weight. These are metals. These are non-metals also, both metals and non-metals, as you can see, about a dozen such elements are there, and these elements actually play a great role in biological systems. So, together I call the body as the fatty when you have carbon, hydrogen, oxygen, nitrogen, and the strong when you have a calcium, sodium, potassium, magnesium, phosphorus, etc., chloride, these things, and then I would say healthy when you have these trace and ultra trace elements present in the body. And that is how we can probably, from the point of view of the inorganic chemistry of life, we can classify this into these kind of aspects of it. So, all this, so many other things I explained, I cannot go into everything, but I will only take to the highlights. So, having talked to you about the bulk elements and the strong, the heavy elements and the elements which are present only in the trace and ultra trace elements, which are very healthy kind of elements, you can see the periodic table. So, the periodic table where the star ones are all the ones which the enzymes are also known. So, for example, in the transition metal, titanium is known as some kind of a nutrient, but still is not clear whether any enzymes, there is no enzymes. Vanadium is okay, chromium also no enzymes, but chromium is a micronutrient. Then manganese, iron, cobalt, nickel, copper, zinc, all of them have got enzymes. Molybdenum also has enzymes. Tungsten is similar to that of the molybdenum, so therefore, I will not be treating with this, and this is found not in human systems, but in microorganisms where you have the microorganisms which grow at very high temperatures like lava, etc. So, therefore, I have given it up. So, I have also talked to you in this particular course about the mercury detoxification, etc., and there are some other essential elements. These are the bulk ones too. Okay. And the main thing what you can say, so barring some exception below, this is the line where you have the nature has chosen the biological elements above this, but not so much below this, barring these ones. So, this is not clear. We try to, in the class I have explained you to compare with their abundances in water, with their abundance in soil, and with their abundance in the human tissue, and found it is not easy to make a direct correlation between the elements that are present in abundant in water, abundant in the soil, etc., have not necessarily be a part of the biological system. On the other hand, there are some elements which are not so much present, which are present very low concentrations, but they are present in the biological systems. And there are some where present in both, like iron, which is present in both. Silicon is present in the earth crust, but not present in the human enzyme, but it is as a micronutrient. Still no enzyme has been identified this. So, like that there are things which are there, which are not there. So, therefore, it is nothing to do with exactly the concentration of these elements present in the sea water or the earth crust, rather they are dependent on that ion characteristics like its charge, size, redox potential, and if it is ion is a cation, then its coordination chemistry properties like preferential geometry, stability, thermodynamic parameters, then the lability parameters which involve in the reactivity. So, the sizes, oxidation states, charges, and HSAB characteristics, that is hard, soft, acid base characteristics, and the coordination chemistry parameters, stability parameters, and lability parameters. So, these seem to be more important than the concentration of these elements alone in the water or in the or in the earth crust. That is how probably the nature has chosen these ones. Also, I have highlighted to you these essential trace elements are present to certain extent, below which is a danger, above which is also a danger. So, below which the concentration is called deficiency, above this is called the toxicity. So, the deficiency and toxicity of this. So, as we increase the concentration of the dosage, which are these elements? These ultra trace and trace. Trace and ultra trace on the x axis, their concentration as the dosage, and look at the response on the y axis. So, the response slowly increases and response becomes a plateau, and this whole range is good enough. Beyond that, if you have more concentration of the ultra trace and trace, then there is a toxicity, and toxicity can lead to death also at a very high concentration. So, that means another condition that the life has imposed on these elements is the trace and ultra trace elements must be, concentrations must be maintained in the body. They cannot be afforded to have very low, they cannot be afforded to have very high concentrations. In both the cases, body will have a problem in terms of its physiology, and that is what we have. So, it is very essential. So, therefore, this slide and maybe one or two more slides will show you the dosage, and the deficiency gives some problems and excess will give some problems like copper, iron, these are the things that you will know. So, again some more elements, the dosage and deficiency syndrome and excess syndrome. Okay. So, this is a very essential aspect to be kept in mind. So, because we cannot take as much amount as available or something to the biological systems, because their excess will cause a problem, their less in the concentration will also cause a problem on this. Okay, let us look at another aspect of highlight that I talked to you earlier is the absorption. So, all these elements we are talking about their presence in the body, but they have to be absorbed. So, example is taken for iron here, iron for exterior into the lumen. Lumen has got certain kind of a complex ligands which will exchange and form their chelates, and these chelates can pass through the intestine. Again in the intestine, the ions can be exchanged. This iron is not neutral iron, these are the ions, and they form the chelates, the endogenous ligands, and this will be taken into the blood. In the blood, these chelates are being further transferring their ion to the transferrin, reticulocyte cells, etc. So, this is the kind of a general mechanism, and the mechanism very well suited for iron. Similarly, many ions will have their own mechanisms. So, with all this, what I have tried to conclude at that stage was that if you take this as the metalloprotein and the metal ion center, you blow this and see this metal ion center has a tyrosine, another histidine, another aspartic, and these four are bound like a kind of a coordination, not like the coordination like the tetrahedral, which is basically of course distorted tetrahedral. So, therefore, the metal ion maintains a kind of a geometry, maintains a coordination sphere of primary, and then maintains then surrounding that is the protein. So, therefore, a metalloprotein, nothing wrong in visualizing as metal ion, a metal ion coordination plus the protein of this. And so, whatever we have realized, understood in the general elements in the biological systems is that visualization of the elements or the ions of importance in biology. So, a metalloprotein is a metal ion plus the protein surroundings. So, when the metal ion is present, it is not just a free metal ion, it can be coordination complex of the metal ion by the protein. Now, in this context, for an inorganic chemist, the protein is a ligand, and for a biochemist, the inorganic center is just one element. So, there are the two kinds of things that one would look at the same. Okay, it is like looking at the half filled glass. Somebody says the glass has a half filled water, and somebody else will say the glass is half empty, and this is the kind of a situation that you will look at. So, metalloenzyme therefore is a metal ion plus protein which binds to it plus exhibiting a function. So, metalloprotein to metalloenzyme is the function added. So, we have taken care of all these things, what does elements do in the biological systems and which of those are important in the periodic table, etc. I have already explained. How are these elements absorbed, I have explained. What concentrations, what happens if the concentrations have gone up, what happens if the concentrations have gone down. Okay. So, how are these bound? All these kinds of things I have already addressed. Just let me tell you how these are bound once again as a highlight. A few of the amino acids are shown here with the carboxylic terminal or phenolic terminal or hydroxy terminal. These are the three types, and here imidazole terminal or arginine terminal or lysine terminal amine, or you have here sulfhydryl function. Methionine, there is an error, it is SCH3, there is an error here, this CH2 SCH3. So, methionine. So, you have thiotype type or these ones or these nitrogen ones, oxygen kind of things. These are directly coming from the side chains of the proteins, and besides these ones, the side chains the proteins are very common. There could be carboxylic, there could be nitrogen based, there could be sulfur based. Besides that, there are some special motifs are present in the biological systems like porphyrin shown in the first one, chorin, the difference is here, this is being saturated, the conjugation is reduced, and chlorine again conjugation is reduced here as well. So, therefore, you have a porphyrin, then chorin and chlorine, etc., and factor 430, all of these have a ring like structure with 4 nitrogens. Therefore, 4 nitrogens can be bound to the metal, then you can have 5th and 6th R can be filled by other systems. There are a few small molecules also are involved, like this one, which is for the iron, which is enterobactin. It is an iron carrier in the lower organisms, where this kind of a compound is oozed out into the medium. The medium, it picks up the ions and swallows back. So, therefore, the ion is coming inside. There are some non-cyclic ones are also there. So, therefore, these are for some for sodium, some for calcium, etc., all of these kind. So, these are referred as the ionophores. So, the binding is one to the side chains, also to the some special motifs, and there are some ions are transported by the ionophores as well. So, overall what we can say, this vanadium, manganese, iron, cobalt, nickel, copper, zinc, etc. These are all for show variety of functions like oxygenation function, halogenation function, ATPase inhibition, and nitrogen fixation in case of vanadium. And manganese, several oxidative things, superoxide dismutase, dehydrogenase, as well as photosynthesis. So, important thing is photosynthesis, and I have explained all of these under the story of manganese. Then when you come to the iron, you have oxygenase, deoxygenase, oxygen transport, the iron transport, electron transport, oxidative reactions, reductive reactions, oxygenation, reduction, reductases, superoxide dismutase. And ribonucleotide reductase. So many things we have already looked at all these. I will be bringing some highlights anyhow. Cobalt case, it is mainly the cobalt in the form of vitamin B12, and which does the group transfer reactions. In the nickel, we have looked at the urease, which is a hydrolytic one. Then we have looked at the hydrogenase, we have looked at the dehydrogenase enzymes too. Copper again is another enzyme where too many proteins are there, too many enzymes are there. So, oxidase, so it will be oxidase, dioxygen transport, electron transfer, So, oxygenation, superoxide dismutase, variety of things are there, and zinc is also though it is a non-redox metal ion, the kind of reaction that it does is the total spectrum. So, you have a hydrolyzed kind, oxidoreductase kind, transferase kind, lipase kind, ligase, all of these kinds of things. Then the molybdenum is nitrogen fixation most important, then oxidoreductase, which is basically a part of the oxotransferase, the process in it. Then you have sodium, potassium, magnesium, calcium, which are all charge carriers, osmotic balance, and magnesium is can do hydrolase, structure, isomerase. Calcium, when it comes, the trigger functions also there, and so all of these kind of functions I have tried to highlight. This is the thing, one part of the introductory aspect that I have covered based on the elements present in biology. Then I have quickly explained a little bit on the biological molecules, protein synthesis, that kind of thing. So, proteins are governed by primary structure, which is the amino acid sequence, secondary structure, which is alpha helical beta sheet, tertiary structure. All of these are integrated into the quaternary structure. Such tertiary structured systems are joined together to form a quaternary structure. Okay. So, that is where we have looked at. So, here you see this is the sequence, this is the primary structure, this is one of the alpha helix, it can be beta sheet also secondary structure. So, all of these are integrated into the polypeptide chain, it is called tertiary structure. Such tertiary structures are present 1, 2, 3, 4, it is called quaternary structure. I also looked at the DNA. DNA is nothing but phosphate, sugar, phosphate, sugar, phosphate, sugar, and the sugar case 3 prime, 5 prime sugar, and the phosphate, phosphate, phosphate. And then on this one at the 2 prime, 1 prime sorry, 1 prime of the the glyco moiety or ribose moiety, you have your nucleic base. So, therefore, nucleic base comes as a side chain. Now, when this forms a kind of helical structure, you can see that the nucleic bases are coming like this, etc. These three different kind of DNAs, aDNA, bDNA, zDNA. If you look at that, their structural morphology are different. They are stabilized by certain hydrogen bonds called complementarity, complementarity of the residues in terms of their hydrogen bonds, T with A and C with G. Okay. So, and these are the different kinds of things. So, you have a grooves like a major groove and a groove like the minor groove this side. So, major groove this side and minor groove that side. So, these are some of the things where molecules can bind. Also, they can also disrupt this the nucleic base stacking and enter too, that is also possible. Now, some important aspects of the biochemical thing is that the protein synthesis is basically from the DNA you have replication, and then that is being reformed by the process, the mRNA, then that is being translated. So, this transcription, then mRNA, then translation, then gives the protein kind of thing. This is basically referred as a central dogma of molecular biology. So, after the protein is synthesized, protein will start maturing. It is called makes into the secondary, tertiary, quaternary structures, that is called the protein maturation. So, basically protein becomes the structure. So, when will the metalloprotein form? So, metalloprotein forms after the protein is formed only in various ways. So, you have a protein as a polypeptide. So, polypeptide can make into its kind of a folds, and these folds can be without the metal initially, and the metal ion can be inserted and the folds get stabilized, and the further that stabilization, instead of folding you can use stabilization, you can get the regular holoprotein. So, this is the holo enzyme or holoprotein. So, this is metal bond. This alternate way is this mRNA, and then you have a polypeptide. You can introduce the metal ion prior to the folding, and this will make into the folding, and it can also form. So, metal insertion can happen before folding or after folding, it is possible. So, the folding will give this one, the metal insertion will give that one. So, and the reverse for the release of this. So, these are the kinds of things. That means, when you take a holoprotein and remove the metal ion, some of these things are possible to go back to the unfolded state, etc. So, and when you have a original protein plus the metal ion holoprotein, there may or may not be some changes, but there is possibility of some changes in the protein conformations too. And then I explained because see, we were looking at in the metalloproteins and metalloenzymes, metal ion is there. Now, you can replace the metal ion by another one, then you can see whether the function is working or not working. On the other hand, if you want to see the kind of a primary residues that which are bound to that, and you want to know whether a particular histidine is definitely essential or not for this metalloenzyme to function, what you need to do? You need to trim or replace that particular histidine by some other amino acid which is not a histidine, and that is what is done by site targeted mutagenesis. So, you have a genomic DNA, you have a gene, and this gene can be put into the plasmid by cutting the plasmid and then put that one. So, you have done in plasmid, and this plasmid you express now. So, when you express this now, you get the same protein with whatever the modification you have done. You have done single mutation, and only in one place you have an amino acid being different. So, for each mutation you have 3 codons are there, that is a codon 3, the nucleotide centers are present, and that is what the thing is. So, these are some of the important ones. So, coming to the understanding of the metal center, as I said, the metal center in the metalloenzymes are very important in terms of their binding, their coordination number, nature of the coordination, because the certain of these things are dependent on the HSAB principles and their geometries also. So, geometry can be linear, can be triangular, can be tetrahedral, can be trigonal, bipyramidal, octahedral, prismatic, pentagonal, bipyramid, trigonal. So, mostly transition metal ions will be showing 4, 6, occasionally 2, but very rarely 7. So, that is why I have given the geometries up to here and some examples too. And in the coordination chemistry, we have studied something called crystal fields theory, which I have not taught in my course, but I presume that you are aware, and try to look at. So, these are all the weak ligands which will split the energy levels or the d energy levels under the let us say octahedral field as T2GeZ. These will split to a less extent as you go towards the right, further and more splitting, more and more, and extreme splittings come in cyanide and carbon monoxide kind of a ligands, or methyl ligand or phenyl ligand, etc. These are all anions kind of things. So, carbon. So, these side are the weak field, these are called the strong field, and there the difference 10 delta Q is very small, little bit more, and large, greater. So, how do one explain this? Obviously, so this is a very important thing. Many times people tend to explain this by the electronegativity principle, which is definitely not correct. You have to invoke the bonding character, and in the bonding it is not the sigma bonding character, it is a pi bonding character. So, those which are pi acidic more of it, and they are in the higher side. Okay. So, therefore, pi acidic character increase from left to the right. Some of them may be pi bases, and then not so much pi and pi acid kind of thing. So, that is how we can make that. So, overall the coordination chemistry parameter that is required to understand the role of inorganic elements in the as a coordination sphere in a metalloprotein or metalloenzyme is their chelate effects, their polarizabilities, hot soft acid base characteristics, which you know very well, I am not going to explain again now, and their stability and their reliability aspects of it. And I have tried to explain the stability, as you see from the Irving-Williams plots, how they are there depending upon the ligands, their things will be, and keeping the metal ion constant, changing the ligands, keeping the ligand constant, changing the metal, you can try to find out the stabilizations of it, and these will follow the HSAB kind of a principle. On the other hand, the metalloenzymes, they are not only metal ion is present and then very stable coordination sphere is there, they also do some function that is called lability. So, here the lability is given, lability rates are given based on the water exchange reactions. So, these are based on the water exchange reactions. So, So, in the water exchange reactions, how fast an outside water will get exchanged with the inside water. So, as you see in a given group like lithium, sodium, potassium, as the heaviness increases, the lability increases. Similarly, beryllium, magnesium, calcium, etc. And if you look at these from compared to monocations, the dications are less labile, trications are somewhat less labile, tetracations are much more less labile. As the heaviness increases, the lability increases. As the charge increases, the lability decreases. And of course, in case of the transition metal, what are you have to see? You have to see not only this, whether they are low spin or high spin, because the low spin and high spin their sizes will vary, their spin density will vary, and that is the reason why we are looking at all of this. So, in effect what we are saying is stability means formation of the complex means delta G is minus NFE. So, delta G minus NFE, if the potential is positive, minus NFE negative, is feasible. If the potential is negative is positive, is not feasible. On the other hand, lability means exchange of the reaction means this one delta G dagger, double dagger, this is the crystal field stabilization energy of the reactant minus crystal field stabilization energy of the activated complex. If it is positive, it is labile, if it is negative, it is basically inert kind of thing. So, therefore, what we have learned, in metalloproteins, the metal ions can bind to the side chain upon deprotonation, they can bind to the and form a coordination complex. In some cases, they can be bound to the special cores like porphyrins, etc., and these things. And how do we understand that it is binding to the side chains? Polarization of the group. It could be a carboxyl group polarization, water group polarization, or imidazole group. Example is shown here. When the metal ion is bound to the water and the pK of the water will reduce, it can go from 14 to 10 and going from no metal to the zinc. Similarly, imidazole on going from the no metal, which is 6 to about 4 when you go to the nickel or copper. So, therefore, in presence of the metal ions, all these can be polarized and they are all bound. So, with this I have been able to explain you the highlights of the metal ions, their roles, biological roles, and the existence as a coordination complex, therefore, their existence, their thing as a stability, and since they are functioning as a lability of it. And I will continue with this revision part in the next class. Thank you.